Okay, this is a brief talk about Mendel's next postulate, that is that traits assort independently. The idea here is that we're looking at multiple traits. Now, whenever you cross P's, of course, there are many traits that are changing over the generations. That doesn't change. The difference between a monohybrid cross and a dihybrid cross, which is what we'll be evaluating next, is that in a monohybrid cross you just care about one of those traits. Tall versus short plants, wrinkled seeds versus round seeds. In a dihybrid cross, you actually follow the inheritance of multiple traits. So when you do that, you have to ask yourself the question, and Mendel asked himself this question: how will these traits assort? Will they assort together or separately? In this particular slide here, we have a yellow round seed and a green wrinkled seed. These are going to be the P generations, or plants with these seeds are going to be in the P generation. Now, the yellow round seed is that's exhibiting both dominant traits, the yellow seed color and the round seed shape. And the green wrinkled seed is exhibiting both the recessive traits, the green color and the wrinkled shape. Alright, now the question is, when you cross these plants from the P generation, how will the traits assort? How will the traits be inherited? Well, going from the P generation to the F1 generation, basically, each of these plants can produce gametes, and each of them can actually only produce one type of gamete. The yellow round, the plant with the yellow round seeds only produces big Y, big R gametes, and the green wrinkle plant only produces little y, little r. And so in the F1 generation, you get what we call the double heterozygote, which exhibits the dominant trait in both cases. Now, when you cross those together, if you kind of expected the traits to assort together, what would happen is then what I mean by assort together, it's like they're kind of linked, so that yellow always is inherited with roundness and green is always inherited with wrinkleness. Well, so you would expect a 3-to-1 ratio, much like in the monohybrid cross. Well, as you might imagine, the reason for this big red X here is that's not what you observe. What did Mendel observe, and what kind of conclusions did he draw from that? Alright, well, here's what he observed, and I'll zoom in on this so you can see it a little bit better. In the P to F1 cross, you get the double heterozygote, no surprise there. But when you cross the F1s together, or allow them to self, you actually get a variety of results. You get 4 different phenotypes in that cross. You get all combinations: yellow and round, yellow and wrinkled, green and round, green and wrinkled. And to calculate what proportions those phenotypes would be in, you can conduct do a Punnett square. Let me show you how this Punnett square is constructed. The eggs, or I guess the ovules of the F1 generation, there will be 4 different types. What happens is these alleles assort independently. That's the whole point of this chapter. So you have 4 different types of gametes: big Y, big R, little y, big R, and little y, little r, in equal proportions. Same for the sperm or pollen, as how you'd say it in plants. And so when you bring those together in a mating, you just combine the alleles of the genotype, and then you can determine the phenotype just by looking to see which dominant traits are present, and if there are no dominant traits, then the recessive trait is exhibited. And so when you do that, you get and I'll move over here just a little bit, you get this 9-to-3 to 3-to-1 ratio among the phenotypes. And so this proportion, or this ratio, is very important to remember. And moreover, every student class should be able to generate this entire cross from nothing, ex nihilo, without just copying it down from one of the slides, but you should be able to do it yourself. And there's actually some kind of pleasure gained from filling out the genotypes in this big Punnett square. So give it a shot. So in independent assortment, remember this 9-to-3 to 3-to-1 ratio. Now, where does independent assortment actually occur? It occurs in meiosis. We'll talk about meiosis in our next chapter in much greater detail. But as you recall, or as you may recall from ninth grade biology, we've got interphase, prophase, metaphase, and anaphase here illustrated in this slide. And remember that the anaphase is where segregation occurs. This is where each allele in the parental cells goes off in a different direction, segregating to become, only one in the gametes. Now, independent assortment actually occurs here in metaphase, or metaphase and anaphase, I guess you could say. The idea here is that, let's say that each of these chromosomes came from a mother or a father. Let's say the blue ones came from dad and the red ones came from mom, of the parents of the individual here that's now producing gametes. And basically, these chromosomes can line up in either way, either with red on top or blue on bottom. Red on top and blue on bottom, or blue on top and red on bottom. And that will determine which way the alleles eventually go. And since each of these can line up in either way, you can have any combination of alleles moving to the gametes, and so they're not inherited together. The two alleles are not inherited together. They're on different chromosomes, and each of the chromosomes can move in different ways. Okay, I hope that was clear, and this is now the end of the little independent assortment talk.